All right, 10 minutes is up. Does anyone need more time? Okay, I can give everyone an extra 2 minutes. All right, so it doesn't matter if you didn't get to answer all of the questions because we'll go over them now. So this is an example of what I would have highlighted. This first paragraph is just explaining how conjugation works, but I highlighted a portion of the bacterial chromosome because that's the key feature of HFR conjugation. The whole point of HFR cells is that they transfer a bit of the host chromosome to the recipient chromosome, which is how new genes are introduced. I next highlighted the order of genes because that's the point of the experiment, is to determine the loci of these genes. I highlighted gram-negative and facultative because they are characteristics of the bacteria, which I could be asked about later. And let's pretend I don't know what oxytrophic means. I'll still highlight it because I could also still be asked about it. I highlighted the amino acids and glucose because those are the molecules being given to the bacteria. And then I also highlighted colonies B and E because experiment 2 is referring to these specific colonies. And glucose is added in there because the plate only has this one nutrient. Finally, I highlighted serine negative, alanine negative, and tyrosine negative because that's the genotype of the F- bacterium. Does anyone have any questions about my highlights?
I was going to ask you a question in regards to do you just solely highlight or do you take notes? Because I remember when I was using TPR as the book set, I read somewhere that like for each paragraph, like you should take notes, like a sentence like cars or like a short little phrase of like 3 or 4 sentences. I just want to know if you still do that.
I don't, when I write notes on bio passages, I usually write shorthand notes, but I don't summarize like passages or anything. And The only notes that I would write would be like mechanisms or diagrams. In this case, I wouldn't write anything except for unless I'm like trying to answer a question, which I'll explain that later because on one of the questions I did write like a little note thing and I'll explain that when we get there. But I don't really recommend summarizing paragraphs. What I would do is, again, like if a passage is really complicated and just really detailed, I would just write um, like the mechanism or like the overall thing so that I would have that visualization when I get to the questions. Does that make sense?
Yes, it does. Thank you.
Okay. So does anyone want to explain their answer for question 1? Okay, so for this question, we have to go back to the data from experiment 1. We notice that colony G was able to grow on all plates except for plate 5, which lacks tyrosine. This means that colony G doesn't have the enzyme that makes tyrosine, so it's tyrosine negative. If you're confused about the nomenclature, the plus superscript means that the bacteria has the enzyme to synthesize this amino acid. If it's a minus superscript, then it lacks the enzyme and needs to be given this amino acid in whatever media it's in. So colony G is able to grow in plates without additional lactose, serine, and alanine, meaning that it can make these nutrients on its own. So A is our answer. Was anyone confused about this?
Yes,
Or did
I was.
anyone have like a different approach?
Could you like repeat it
Yeah.
again? I'm sorry.
Yeah, so to figure out the genotype, we have to pay attention to the plate in which colony G wasn't able to grow. So that's why we pay attention to plate 5. Colony G wasn't able to grow on plate 5, and to figure out why, we look at what the plate contains. It only contains serine, alanine, and glucose. And because it wasn't able to grow on this plate, that means it's missing something essential. So it's missing this amino acid tyrosine. And if tyrosine is not given on the plate, like in plate 5, that means it's not synthesizing tyrosine on its own. So therefore, it either needs to synthesize tyrosine on its own by getting this gene through conjugation, for example, or tyrosine should be supplemented to the media for it to grow. Does that make sense?
Yeah, that makes sense. And basically, you were able to understand that tyrosine was the missing one because you compared that plate 5 in comparison to the other plates.
Exactly. So you see that colony G was able to grow in all these other plates that are each missing either lactose, serine, alanine. And because it was able to grow in the absence of these, then you can conclude that it does carry the enzyme to synthesize these nutrients.
All right, thank you so much.
No problem. All right, so we'll move on to question 2, if everyone's okay with that. Okay. Okay, so the definition of an auxotroph is exactly what the question is saying. It's a mutant strain of bacteria that requires additional nutrients that a wild type bacteria is able to make. So the definitions of the other terms listed, like chemotroph, heterotroph, autotroph, as well as phototrophs, are simply classifications of bacteria according to where they get their energy and organic molecules from. So chemotrophs get their energy from chemicals. Heterotrophs use organic molecules from other organisms to synthesize their own nutrients, and autotrophs use CO2 to build their organic molecules. Was everyone okay with this question?
Yes.
Okay, great. So I highlighted the characteristics of the bacteria on, oh wait, no, sorry, I'm on the wrong question. Okay, so for this question, we have to take a look at plate 4 from experiment 1. This plate is given alanine, tyrosine, and glucose. So it's missing serine. Only strains of bacteria that are able to synthesize serine can grow on this plate. And since it's not given on the plate. So if the genotype is serine negative, then the bacteria cannot grow because it needs this amino acid, whether it be from synthesizing the amino acid itself or being in a plate that already has it. So notice how we only need to pay attention to serine for this question. The strain can be alanine plus or alanine negative or tyrosine negative or tyrosine plus. It doesn't matter because the plate already has these amino acids. The bacteria will only need to synthesize serine. So when you're given a question like this, start by what is given on the plate. If there's no growth, then the bacteria is clearly missing something essential. So for example, let's say protein X. If protein X isn't given on the plate, then the bacteria is not able to make protein X, which is the reason why it's not able to grow. Does that make sense?
Sorry, so for this question, you said that like it's going to be serine positive because we know that positive that stands for it has the enzyme, so it's going to be able to grow.
Yes. So in order to grow in this plate, it only needs to synthesize serine. So the genotype has to be serine plus because the plate doesn't have serine.
Thank you.
No problem. Okay, so if everyone's fine with the previous question, is it okay if I move on to question 4?
Yes.
Okay. So like I said, I highlighted the characteristics of the bacteria in the passage. Remember how I highlighted gram-negative and facultative? I don't know how to pronounce that. However, if I were in the real exam, I would easily be able to go back to that highlight and use that information to answer this question. So the passage said that E. coli is a gram-negative facultative anaerobe. Gram-negative bacteria has 2 layers of cell membrane and it has a thin layer of peptidoglycan sandwiched in between these 2 layers of cell membrane. Because of this, the violet dye will stain gram-negative bacteria weakly, and so the bacteria will look pink. So E. coli will stain pink. And facultative anaerobes will use oxygen only when it's available, but they don't need oxygen for its survival. So did everyone get D for this question? Okay. Yeah, so in the November 12 topic session, I explained like more about gram positive and gram negative and sort of like an easier way to distinguish between them. So when you're staining bacteria, you're always using a purple dye. So I thought gram negative is negative for this purple dye, so it'll stain pink, and gram positive is positive for the purple dye, so it'll stain purple. I don't know if that will help you. It helped me. Yeah. Okay, I hope that helps. And I'm sorry if I confused you more. Yeah, so the way I thought of it was when you're staining bacteria, you're always using a purple dye. So gram negative will be negative for the purple dye, so it'll stain pink. And gram positive is positive for this purple dye, so gram positive bacteria will stain purple. Let me type it.
Sorry to interrupt. My Zoom disconnected, but just to clarify, if it's gram negative, it stains pink?
Yes.
Okay, and then what about the oxygen part?
So again, I explained more about that in the topic session, but for the oxygen part, there's 4 types. There is obligate aerobes, which require oxygen for survival. There's facultative anaerobes, which will use oxygen when it's available, but they don't need this.
Okay.
Tolerant anaerobes will tolerate oxygen when it's present, but they won't use it. So that's the difference between facultative and tolerant.
Okay. And did the
And then
passage,
there's,
sorry to interrupt, did the
Yeah, sorry.
passage explain that E. coli was facultative then? Or did we have to
yes,
know
in the
that?
passage, no, in the passage it said E. coli, comma, a gram-negative and facultative anaerobe.
Okay, perfect. Thanks.
So in experiment 2, which part? Wait, do you want me to go back to the passage? Yeah, so here under experiment 1, I wrote E. coli, gram-negative and facultative anaerobic bacteria.
No, like you
So
said,
that's why,
sorry, you said
Yeah.
that when staining, you use purple dye for this. Can you repeat that? I didn't quite get you.
yeah, so when you're staining for bacteria, you'll always use a violet dye because peptidoglycans it like retains that purple dye. So the way I thought of it was gram-negative is negative for this purple dye, so it'll stain pink. And the reason for that is because the dye can't stain peptidoglycan because it has this outer membrane covering it. And gram-positive bacteria, they have a thick layer of peptidoglycan, so they don't have this outer cell membrane. So that's why gram-positive bacteria are able to stain violet very strongly. So that's why I made up like a little mnemonic, like gram-negative is negative for the purple dye. Gram-positive is positive for the purple dye.
Okay, thank you.
No problem. So in experiment 2, bacteria from colonies B and E undergo conjugation. The question is asking us to determine the genotype of the bacteria that has genetic information from colonies B and E. To do this, we have to determine the genotypes of B and E by looking at data from experiment 1, which I have here. We noticed that bacteria from colony B are not able to grow on plate 4, which lacks serine. Therefore, colony B is able to synthesize alanine and tyrosine, but not serine. So this is an example of a note that I would write during the exam. I would write colony B, the genotype for colony B, which is serine negative, alanine plus, and tyrosine plus. Colony E is not able to grow in plates 3, which lacks alanine, and plate 5, which lacks tyrosine. This means that colony E lacks the enzymes needed to synthesize tyrosine and alanine. So on my noteboard, I would write alanine minus, tyrosine minus for colony E, and serine plus. And when these two colonies undergo conjugation, the resultant or mixed bacteria will have a combination of their traits. So because colony B can make alanine and tyrosine and colony E can make serine, the mixed genotype will be serine plus, alanine plus, and tyrosine plus. Does that make sense?
Yes, it does.
Okay, was anyone like confused about this question? But yeah, this is what I mean by like writing these shorthand notes helps you like understand the question better. Okay, so we will move on to the final question. To answer this, we need to refer back to the data from experiment 3. The way I like to answer questions is, like I said, by visualizing them, especially if they're experiment-based. So this is what this setup would look like. You have plate 1 with serine and alanine, plate 2 with alanine and tyrosine, and plate 3 with serine and tyrosine. The passage states that the purpose of this experiment is to determine the order of genes transferred, which is what I highlighted. So conjugation is allowed to continue for 5 minutes and then it stops. The scientist observes the plates and sees that there is growth on plate 3, which contains serine and tyrosine. The fact that there's growth indicates that the bacteria is able to synthesize the third missing amino acid, which is alanine. This means that the gene required to make alanine is transferred first. Does that part make sense? Okay, so right now I'm just explaining like what the experiment setup looks like. So like I said, you have plate 1 with serine and alanine here, plate 2 with alanine and tyrosine, plate 3 with serine and tyrosine. So following this data, we stop conjugation after 5 minutes. And there's only growth on plate 3 with serine and tyrosine. So 5 minutes after conjugation means that some genetic information was transferred. And we determined this by looking at plate 3 because there's growth here. The fact that there's growth means that, yeah, the gene required for alanine synthesis is transferred first. Okay. So, okay. The scientist now allows conjugation to continue again for another 5 minutes and then he stops. The scientist now observes growth on plate 2, which has alanine and tyrosine and lacks serine. The bacteria on this plate are only allowed to grow because the bacteria is able to synthesize serine on its own. It's able to do this because of conjugation, so genetic transfer. And the fact that it's able to grow after 10 minutes means that the gene to make serine is transferred second after alanine. Does that part make sense? Okay. Finally, after 15 minutes, the scientist observes growth on plate 1, as we can see in the table. Again, plate 1 contains serine and alanine. So the only way for the bacteria to grow on this plate is if it's able to make tyrosine on its own. The fact that there's growth after 15 minutes, way after the other plates have shown growth, indicates that the gene that encodes tyrosine is transferred last. So this is a visual representation on the left of the F plasmid in purple that has integrated into the host chromosome, which is in red, like the passage describes. Like I said, HFR cells are useful for mapping genes. I went more into detail during the topic session last week. But the most important thing to take from this picture is that relative to the F plasmid and the origin of replication here, which is the arrow, we see that the gene for alanine occurs first. The squiggly line represents where conjugation got interrupted, so after 5 minutes. At the 10-minute mark here, the gene for serine is transferred. And at the 15-minute mark, the gene for tyrosine is transferred. So that's why scientists will often stop conjugation at like short intervals so that they can see which genes are transferred first based on the bacteria's growth. And the picture on the right is just describing conjugation. Again, I went more into detail on the November 12th session. And if you're having trouble understanding the content, I would suggest watching the recording of this session because I talk about conjugation and mapping towards the end. Does this make sense? Okay, because I will also give you guys an answer key of the passages, and in the answer key, I write my explanations for the questions. Okay, that's it for my part. I'm sorry guys if you got confused in the end. I tried to make this as straightforward as possible, but conjugation and HFR mapping is itself a quite complex topic. So this is as far depth you'll see. So yeah, again, if you have any questions about the passages, please let me know. You can email us. I'll answer any questions that you guys have.
Okay, thanks, Ashley. We're going to do the next passage is, I'll paste the link in the chat, but we also sent out an email. So let me know when you guys are ready and I can start the timer. Let me know in the chat when you guys are ready to start. Okay, I will start the timer now. Does anyone need more time? Okay, cool. We'll go over it then. So for question 1, what did you guys get?
I got C.
Okay, cool. So this is talking about the central carbon in, hold up, let me go back. Right here. So in the, do you guys see this bond right here with the carbon and the nitrogen? So that's a triple bond, and the carbon in the center is linear. So that would be sp. And for anything that is linear, that is 180 degrees. Does that make sense? So this is kind of a summary of what you guys should definitely know for the MCAT, the different types of arrangements and their angles, and how to tell from that. Okay, moving on. For question 2, what did you guys put for this?
I had C, but I wasn't really sure about it.
Okay, cool. Okay, so I'm not sure if we've gone over enantiomers, but those are non-superimposable mirror images of each other. So those, they're just different in one stereocenter. So they actually have the same identical chemical and physical properties. So that would make 1 and 2 correct. Because they would look the same, they would have the same boiling point, they would have the same IR spectrum as well. So, and then one of these is correct. So we just have to figure out which one is correct. So that would eliminate C and D, just because it doesn't have 2 and it has both 3 and 4. And then going from there, the enantiomers would not react with the chiral reagent because they have different orientations. So one is R and one is S. So they would not react with the same chiral reagent. So it would be A. Let me know if that makes sense. Okay, yeah. Cool. Is what true for all enantiomers? It is true that enantiomers have the same boiling point and the same IR spectrum, yes. The rate of reaction with the chiral reagent? Yes, that should be true. Or no, the achiral, because they wouldn't have the same reaction with the same chiral reagent just because they have different, they're enantiomers, so they have different stereocenters, so they would react differently with the achiral reagent. Let me know if that makes sense. Okay.
Sorry to interrupt. So for the IR spectrum, the functional groups, is that what determines the IR spectrum?
Yeah, so there's different functional groups that show up. You're correct. So there's usually the carbon-carbon bond, there's a list of which ones you should definitely know for the test. Let me see if I can pull up a quick chart. But the type of bonds that are important to know for those are like the carbonyl bond, the phenyl group, the alcohol, the OH stretch, And those have different wave numbers and intensity. So that's definitely something we should know for the test. Yep.
Okay, and then I guess because they're enantiomers, they have the same functional groups. So that's why you can say that they're the same.
Yeah, that's correct. As you see in the diagram that they showed, that I can go back to it real quick. That they're basically the same. Do you see that?
Mhm.
Okay, so except for right here where the stereocenter one is on a wedge and here it's on a dash right there, and here it's just switched right here. So, but everything else is the same. So they would show up in the same IR spectrum and the boiling point would be the same as well. But just the stereocenter right here is different.
Okay, thank you.
Oh, okay. Yeah, this is just going over how to tell enantiomers from diastereomers and the differences. So here, these are enantiomers because they have different and opposite stereocenters. So where there's a R, here is S, and here's a R and there is S. So that's a quick way to tell if something is an enantiomer because they would have the opposite. Diastereomers have one that is the opposite, but not all of them. So here, R and S is the opposite, but here we also have one that is the same. And same thing here. Let me know if that makes sense. But that's like a quick way to tell if something is an enantiomer versus a diastereomer. And diastereomers, they have different physical properties.
So just to be clear, you said for diastereomers, they have a stereosensor, just that like one of them is the same?
Like one of them is the opposite actually. Not all of them are opposites, but
Okay, one of them is
yeah.
the opposite. So isn't that, so what would be the difference between the epimer and then the diastereomer?
The epimer is they have all of them are different except for, no, sorry, all of them are the same except for one, the last, here, let me show you. There's just two isomers and with different like atoms, except for one of the asymmetric carbon atom is different. It's easier to show with an example. So let me pull this up real quick. I might have to exit, but. Okay, do you see that? So here, all of these are the same, and then only the last one is different, and that's what the epimer is right here at the carbon center right there. For diastereomers, that's not always the case. You just need at least one that is opposite, but some might be the same, some might not.
All right, thank you.
Okay, what about 3? What did you guys get for this?
I got A.
Okay, cool. Yeah, so here you just have to go through and find out which one is, both of those stereocenters are R. So that means you would want to just like quickly like rearrange both of them and see which one are rotating clockwise. So here, the first one would go to the oxygen. Here, let me write this down. So our first priority would be at the oxygen, and then second would be at this carbon right here. And the third would be at this center right here. So as you see, we're going around clockwise and the hydrogen is already facing away. So this is R. And then same thing for this carbon. our first priority would be this oxygen, and then second this carbon, and then this carbon right there. So this is going counterclockwise, but the hydrogen is on a wedge, so we have to flip this. So since it's going clockwise or counterclockwise, the actual rotation would be R. Does that make sense?
Yes. And then could you repeat what the dash means? I always get it confused. Is it like away or
Yeah. So dashes mean it's going away from you and wedges are facing toward you. So when it's going away, that's fine. And you keep the orientation that it kind of tells you. So since a hydrogen, you want the lowest priority away from you. Hydrogen is already on the dash, so it's fine. So you can keep this as R. But here hydrogen is facing towards you and it's not on a dash, whereas the oxygen is. So you kind of have to flip those. And an easy trick is to just take the opposite. So if we got S here, because it's counterclockwise, so we would just flip that and the S would go to a R.
I was wondering, is there a way to do this that's kind of quicker? Because I find that these questions tend to take up a lot of time when I'm doing passages.
Sorry, let me think real quick. I usually just end up doing this, but because I have a lot of practice, but for R, since they're the same, you would have to find both of them on the, actually, sorry, hold up, you flipped this.
I know like a cool trick for, I don't know if you guys know this, but like you can, to remember R and S, like if it's 1, 2, 3, like this one is counterclockwise, so it'll be like S.
Yeah.
But like since this is like you have to reverse it because this is the lowest priority. I mean, this is like all the way in the back, so then it would be R. And if it's clockwise, then it'll be like R.
Thank you. That's great.
Okay, no problem.
Yep, I think like what really helps is just to do a bunch of these with practice and remember those tricks and stuff. Yep. Okay, so what did you guys get for 4?
I said B.
Okay. So to separate these diastereomers, we have to, this is a crystallization process where first you have to heat up the solid to dissolve it. And then you cool it down slowly. And then once it cools down, it forms a precipitate. And then you can use a cold solution to obtain the precipitate and the crystals. So that is actually referring to C, which means you would first heat up the solution and it dissolves. So once it dissolves and you heat it up, then you cool it down. And then at room temperature, once it's cooled down, then it starts appearing as a solid. Let me know if that makes sense. Okay, so which part are you confused on?
I guess, so you explained the crystallization process, but how does that relate to the solvent choice?
Right, well, if both of them are, so when it's heated, there's a hot water bath and there's a room temperature. So in the hot water bath, there shouldn't be a solute because it should be dissolved at a higher temperature. So no solute should be visible. And then when it cools down to room temperature, that's when you should start seeing the solute. Let me know if that makes sense.
Yeah, that makes sense. Thanks for
Okay.
clarifying.
So for
Okay.
question 4, I'm sorry.
Oh, no worries.
Would you say that basically to answer this question, you would just have to really know what the crystallization process is? Like that would be the content.
Yeah, and it mentions it in the experiment, the solubility results for the crystallization experiment. Yeah, that is something you should know.
Thank you.
Okay, last question. What did you guys say for this one? Okay. So it mentioned the log p-values in the, we can go back and see the relationship. But it says non-polar solvents have a higher log p-value, and we're trying to look at this one right here, and the highest one is 4.4. So this is pretty high compared to everything else. So we can go that it's going to dissolve in the organic non-polar solvent. And let me go back. That would mean it is lipophilic because it is going to dissolve in the organic non-polar solvent. And if it's dissolving in that, it's going to have a high volume of distribution. So just going over these terms, lipophilic means dissolving in lipids or fat, whereas the usual one we hear is like hydrophilic or hydrophobic, which is pertaining to water. So this is kind of the opposite. So lipophilic would mean that you like lipids and like fat. And they mentioned that a log p-value would dissolve in a nonpolar solvent. So that's why it would be lipophilic, and then we would get rid of C and D. And then if it's dissolving and it's lipophilic and it can distribute into the fatty tissue of the body, that means that it has a higher distribution of volume. Does that make sense?
For me, I guess I just don't understand how that correlates to the cytopram's properties. What, like, were we just supposed to look at that and see that it has a lot of like carbons, like
Oh,
molecules? Or...
no, right here it tells you the log p-value, and it says the log p-value of
Oh, okay.
Halofram is 3.58.
Oh, okay.
So yeah, we would use this table right here and look over here for that. And it tells you compounds that dissolve in organic nonpolar solvents have a higher log p-value. So it kind of tells you right here.
Oh, okay. That's why I didn't look at the information given. Thank you.
Yeah, no worries. Okay, so just to sum up with some overall strategies, make sure you just write shorthand notes while reading the passage and highlight those important equations. To really improve in the science section, you want to really understand your incorrect questions and why you got them wrong and learn from your mistakes. And note down like common concepts you missed so you can review those and definitely understand like the graphs and relationships that we talked about today and like break down those passages. Ashley, do you have any other tips you want to mention?
Yeah, so like I said, really important to time yourself from the get-go. Like you want to have that inner clock that tells you if you're spending too much time on a passage that will help you during the exam, because you'll get so caught up with a question and you'll spend so much time with it that you haven't even realized like how much time has been spent. So really important to, even if you're just practicing with like one or two passages, set that alarm. really important to do that. And also, of course, like I said before, really important to take breaks, even if it's just going for a walk or like eating ice cream. Do whatever you can to celebrate all of your studying that you have done so far.
Yeah, I agree. Definitely don't feel afraid to take some time off, especially with Thanksgiving and the holidays coming up. And you guys can do this. And just a reminder, we are volunteers, so if you can, please donate to our PayPal. It will definitely help us become a nonprofit in the future soon. And you can join us on Facebook, and we have a lot of students on there, so you can learn from each other. And thank you for coming today.
Thank you.
Thank you. I was gonna ask, by any chance, like, do you think in the future you guys could make this like a regular thing, like doing passages on on Sundays, based off maybe videos that were like recorded during like during the week on the given topics?
Yeah, thank you for that feedback. We'll definitely try to incorporate more of these in the future. And we'll send out a post survey. And if you want, you can also remind us again on there, and we'll definitely consider doing that.
Thank you so much.
I was wondering if I could actually clarify one of the questions.
Yeah. what's up?
So for the last one, I didn't follow why it was lipophilic versus lipophobic.
Yeah, sure. So it says, do you understand, like, right here, it has a log p-value that is high?
Mhm.
Okay, great. So that means it dissolves in organic non-polar solvents. If something is dissolving, it's not afraid of that substance. I'm not sure if you've like learned like hydrophobic, like scared of water, you know,
Mhm.
and hydrophilic, it likes water. So this is kind of the same thing here. If it's dissolving in the organic non-polar solvent, that means it kind of likes the lipophilic and it's not scared of it. So it's going to be lipophilic and not lipophobic.
Okay, so because it's dissolving in a
Organic, yeah. So organic and non-polar pertains to lipophilic. So those means like your lipids and fats, and that's like non-polar solvents such as hexane and like organic non-polar portions that have a lot of carbons and stuff like that.
okay, thank you so much.
Mhm.